This video is about identification of negative ions. Our objective is to describe how to detect following anions in aqueous solution: chloride, bromide, iodide, sulfate, sulfite, carbonate, and nitrate. Let's start with detection of halide ions. Halide ions are chloride, bromide, and iodide. Test for halide ions. Take small quantity of sample solution. Add an equal volume of dilute nitric acid. Then add aqueous silver nitrate. In this picture, 3 test tubes are shown, which contain sample solutions containing different halide ions. See what happens on adding silver nitrate. As halides of silver are insoluble in water, they produce their respective precipitates on adding silver nitrate. The color of these precipitates are different. The color of silver chloride is white. The color of silver bromide is cream. The color of silver iodide is yellow. Here, we have demonstrated how the tests for identification of halide ions is carried out in the laboratory. 3 test tubes are marked A, B, and C. These test tubes contain salt solutions having one of the halide ions, separately. Then we add aqueous silver nitrate into all the test tubes A, B, and C directly through the reagent bottle or by a dropper. We can add more silver nitrate in the solutions to produce more precipitates. To make sure that 2 solutions are mixed together well, shake the test tubes as shown. You can see the white, yellow, and cream color precipitates, respectively in A, B, and C. So, A contains chloride ions. B contains iodide ions, while C contains bromide ions. Here, ionic equations of these reactions are given, which shows how silver ions combine with halide ions to produce their respective precipitates. Now we will see the detection of sulfate ions. See how we detect sulfate ion. Take small quantity of sample solution. Add an equal volume of dilute hydrochloric acid or dilute nitric acid. Then add aqueous barium chloride or barium nitrate, as shown in the picture. Observations and results. As a result of mixing 2 solutions, one containing sulfate ions, while other containing barium ions, are mixed together, white precipitates of barium sulfate are produced. In this laboratory test you can see how this test is conducted. On adding barium nitrate into the solution, in a test tube containing sulfate ions produces white precipitates of barium sulfate. Ionic equation for the test reaction. Barium and sulfate ions from the solution combine to produce barium sulfate as white precipitates. Now, we will discuss identification of sulfite ions. Sulfite ions can be identified with 2 reagents. One is potassium manganate, 7, while other is potassium dichromate, 6. First, we will discuss identification of sulfite ions with potassium manganate, 7. Test for sulfite ions. Take small quantity of sample solution. Add a small volume of acidified potassium manganate, 7. Potassium manganate, 7, is purple in color. On shaking, the purple color of potassium manganate, 7, discharges to colorless in presence of sulfite ions. Now see the test of sulfite ions with potassium dichromate, 6. Like previous test, take small quantity of sample solution. Add a small volume of acidified potassium dichromate, 6. Observations and results. The color of potassium dichromate, 6, changes from orange to green in presence of sulfite ions. Now, we will see how to identify carbonate ions in a sample. For this laboratory test, first, we take sample in a test tube, either solid or solution. Then we add dilute hydrochloric acid in the sample. Observations. On adding hydrochloric acid into sample, brisk effervescence evolve. These brisk effervescence shows the evolution of carbon dioxide. Test for carbon dioxide gas. Pass the gas through lime water taken in another test tube. You can see bubble when gas is passed through lime water. Pass the gas for a few minutes, then observe. You can see, lime water turns milky. The test indicates the presence of carbonate ions in the sample because carbonates gives brisk effervescence when react with acids. It is due to the evolution of carbon dioxide gas produced during the reaction. While turning of colorless lime water milky shows the evolution of carbon dioxide, which reacts with lime water to produce white precipitates of calcium carbonate. Now we will see how to identify nitrate ions in a solution. During this test, ammonia gas evolve, which is colorless. So, detection of ammonia is also carried out during this scheme. We have two ways to detect evolution of ammonia in the laboratory. We will see, one by one. Test for nitrate ion. Take small quantity of sample, either solid or solution. Add a little quantity of aqueous sodium hydroxide in the sample. Then, add a small piece of aluminium foil, or a little quantity of aluminium powder. Now, gently heat the contents of the test tube. Bring moist red litmus paper near the mouth of the test tube from where gas evolves. The moist red litmus paper turns blue. Inference. Nitrate ions are reduced to ammonia on reacting with aluminium in presence of sodium hydroxide. Thus, turning of moist red litmus paper blue confirms the presence of nitrate ions. You were already told that ammonia can be detected by two ways. One with litmus paper we have already watched in this video, while other is with hydrochloric acid that will be covered now. But how? The first 4 steps describe synthesis of ammonia, which are already illustrated. Take small quantity of sample, solid or solution. Add a little quantity of aqueous sodium hydroxide. Then add a small piece of aluminium foil, or a little quantity of aluminium powder. Gently heat the contents of the test tube. Bring a glass rod dipped in hydrochloric acid. As a result, white fumes evolve. Inference. Nitrate ions are reduced to ammonia on reacting with aluminium in presence of sodium hydroxide. Thus, evolution of white fumes confirms the presence of nitrate ions. So, in this video we have covered the detection of all anions which are included in GCE O-Level syllabus. Watch my other related videos and subscribe. Thank you.